Hello. This module is about the first stage of glycolysis, but includes an overview of energy flow and an overview of glycolysis itself. Then we'll take a closer look at stage 1. We'll look at each reaction and focus on the energetics and enzymes catalyzing each reaction, keeping in mind that glycolysis is the start of a pathway that evolved to get free energy out of glucose. As we move through the glycolytic pathway, we'll do some real-life energetics problems. We'll try to understand the concept of coupled reactions and the role of substrate-level phosphorylation in the production of ATP. We're also going to see that there is some investment of free energy, some investment of or consumption of ATP that is necessary to get glycolysis underway, to get it started. When we study the enzymes of glycolysis, we'll also look at the logic of allosteric regulation of this pathway and the implications of biologically irreversible reactions. Okay, the end game of glycolysis, and in fact of respiration, is to get free energy out of glucose and into ATP. And here's a drawing of the ATP molecule as it would exist at about pH 7, where it is most of the time in cells. ATP is the energy currency of life. Its phosphoanhydride linkages release a lot of free energy when hydrolyzed, which is why we usually call ATP a high-energy phosphate compound, and we call the phosphoanhydride linkages high-energy bonds. The water addition is made easier because the phosphates are very electronegative, creating an effect of repulsion between the phosphates. The hydrolysis of the beta-gamma and of the alpha-beta phosphate linkages release considerable free energy that can be used to do different kinds of cellular work. To understand how this free energy-rich ATP molecule is made, first you may remember that photosynthetic organisms, from cyanobacteria to green algae all the way up to higher plants, capture the free energy of light in chemical nutrients, mainly glucose. All organisms get nutrient-free energy either by fermentation or respiration, putting this free energy into high-energy intermediates like ATP. Then the ATP itself is hydrolyzed in many cellular reactions to harness its free energy to do cellular work for growth or other free energy requiring metabolism. Now let's recall just how much free energy is available from ATP. ATP hydrolysis is very exergonic. The standard free energy change, delta G0, is equal to minus 7.3 kilocalories per mole of ATP hydrolyzed. The dehydration synthesis of ATP is therefore highly endergonic, to the tune of plus 7.3 kilocalories per mole. The free energy needed to do this reaction comes from the fermentation or respiration of nutrient molecules like glucose. Here again is the chemical equation for the combustion of glucose. In cells, this occurs in 3 steps: the principal pathways of glycolysis, the Krebs or TCA cycle, and electron transport. This releases, as you may recall, 686 kilocalories per mole of glucose oxidized. The free energy released in these 3 pathways is captured in ATP, either by substrate level or oxidative phosphorylation. The amount of this 686 kilocalories per mole available from glucose that is actually captured as ATP is about 263 kilocalories. Okay, here is an overview of glycolysis. Glucose, or G here, is delivered to cells where membrane proteins called glucose transporters facilitate their diffusion into the cell. In stage 1 of glycolysis, glucose is phosphorylated, then isomerized to fructose phosphate, and then phosphorylated a second time to make fructose diphosphate. This is stage 1 of glycolysis, in which, as you can see, 2 molecules of ATP have been consumed, and a 6-carbon sugar has been split into 2 3-carbon sugars. Here's stage 2 of glycolysis. Note that each of the reactions in stage 2 happens twice per starting glucose molecule and yields in the end 2 molecules of pyruvate or pyruvic acid. The first reaction of the second stage is an oxidation or a redox reaction involving the oxidation of glyceraldehyde 3-phosphate or G3P. In 2 subsequent reactions, ATP will be made by substrate-level phosphorylation. Because these reactions occur twice, a total of 4 ATP molecules will be made by the time pyruvate is produced. To summarize then, in stage 2 there's been a redox reaction, in fact an oxidation of carbohydrate, and a synthesis of 2 molecules of ATP. Again, each reaction occurring twice per starting molecule of glucose. Well, there are 2 alternative fates for pyruvic acid. In anaerobes, or in cells under anaerobic conditions, pyruvate can be reduced to generate one of several fermentation end products. Two well-known ones are ethanol and lactic acid. At the end of a fermentation, there will have been several redox reactions, but in fact no net oxidation of carbohydrate. On the other hand, in aerobic organisms, or in cells under aerobic conditions, pyruvate will be completely oxidized to carbon dioxide and water by respiration in mitochondria. Now, let's take a close look at stage 1 of glycolysis. Glycolysis begins when D-glucose enters a cell. The first reaction of glycolysis involves the transfer of a phosphate from ATP to glucose. You should recall seeing a computer model of the hexokinase enzyme undergoing induced fit after binding a molecule of glucose. Enzymes that catalyze phosphate transfers between ATP and other molecules are often called kinases. The standard free energy change measured for this reaction, is conducted in a closed system under standard conditions, is minus 4 kilocalories per mole. This is an exergonic reaction. Phosphate transfers like this one are called coupled reactions. Immediately after a meal, glucose enters your bloodstream in the small intestine, usually in large amounts, where much of it enters the liver. That's the first stop for ingested nutrients. The rest of the glucose, once it's past the liver, is distributed to all cells. In plants, photosynthetic cells synthesize glucose from scratch. Inside the cell, glucose is quickly converted to glucose 6-phosphate. Any G6P that is not needed immediately for its free energy would be stored as glycogen in animals or as starch in plants. Stored glycogen is visible in the electron microscope as electron-dense granules, such as those seen in the micrograph in the lower right. Between meals, or at night in the dark in plants, glycogen, or starch, is hydrolyzed to release glucose 1-phosphate, which is in turn converted back to glucose 6-phosphate by the enzyme phosphoglucomutase. Mutases are enzymes that catalyze the transfer of functional groups, often phosphates, from one carbon to another in the same molecule. The fate of glucose 6-phosphate retrieved from storage polysaccharides depends on the cell type in which the reactions are occurring. We'll learn more about this later. Let's look at the energetics of the hexokinase catalyzed reaction. The easiest way to do this is to think of the reaction as the sum of two reactions. The first is the hydrolysis of ATP, shown here. The second is the phosphorylation of glucose, a condensation reaction. We can sum the two reactions by first canceling the terms found in common on both the left and right side of the chemical equations, and then adding the remaining reactants and products as seen in the summed equation here. The hydrolysis of ATP, of course, is very exergonic, which is why the phosphoanhydride linkage in ATP is often referred to as a high-energy bond, as I've indicated earlier. We know that the overall reaction is exergonic, with a standard free energy change of minus 4 kilocalories per mole. After the summing up, we can calculate the free energy changes, and the delta G0 for the phosphorylation of glucose turns out to be plus 3.3 kilocalories per mole. This is a fine example in which an exergonic reaction is back coupled to an endergonic one. Let's take a look at another reaction. What's the standard free energy change for the hydrolysis of glucose 6-phosphate? This is an exergonic reaction, and the standard free energy change is minus 3. It's actually minus 3.3 kilocalories per mole. We'll see that this is an important chemical reaction, especially in liver cells later. Let's look at the enzymatics, I call it, of hexokinase, that is, some of the enzyme's catalytic properties. The catalyzed reaction is biologically irreversible. You'll probably remember from some chemistry course that all chemical reactions, including biochemical ones, are considered reversible. So we call a reaction biologically irreversible when the enzyme does not readily bind and therefore reconvert products to reactants. How does a biologically irreversible hexokinase help an organism? Well, once inside the cell, glucose is rapidly converted to glucose 6-phosphate, and it isn't recognized by glucose transporter proteins that got the glucose into the cell in the first place. So this valuable nutrient, once in the cell, can't get out. Hexokinase is one of the allosterically regulated enzymes of the glycolytic pathway. Glucose 6-phosphate, the product of the catalyzed reaction itself, can bind to an allosteric site on the enzyme and block its catalytic action, in fact blocking its own formation. Glucose 6-phosphate binding at this other or allosteric site causes the enzyme to change shape and to be unable to bind glucose or ATP at its active site. How is allosteric inhibition of hexokinase by glucose 6-phosphate useful to a cell or to an organism? Well, when glucose is plentiful, say right after a meal, it gets into cells and the cells build up high concentrations of glucose 6-phosphate, which then inhibit its own synthesis. Thus, the cells get the glucose they need, leaving the rest to circulate to be shared with other cells and tissues. Let's consider glucose 6-phosphate phosphatase again. This reaction is also biologically irreversible, and the enzyme is only active in the liver between meals. Why do you think that is? It's because between meals it's necessary to mobilize stored glucose, in the liver that would be glycogen, and glucose 6-phosphate phosphatase is part of this process, catalyzing the removal of the phosphate group so that now glucose can cross the cell membrane by binding to the glucose transporter protein, allowing glucose to exit the cell, in this case exit the liver, and be transported to other tissues that need glucose for energy. So here's the rest of stage 1 of glycolysis. In this reaction, catalyzed by glucose 6-phosphate isomerase, the glucose 6-phosphate is converted to fructose 6-phosphate. Remember that glucose and fructose have the same molecular formula, so by definition they are isomers. This reaction, as you can see, is endergonic, but very modestly so. The catalyzed reaction is freely reversible, and I note that enzymes that catalyze the interconversion of isomers are often called isomerases. In the next reaction, fructose 6-phosphate will itself be phosphorylated, and here it is. This exergonic reaction is catalyzed by the enzyme phosphofructokinase, which couples the phosphorylation of fructose 6-phosphate to ATP hydrolysis. Here's the energetics breakdown for the reaction. Once again, the phosphorylation of fructose 6-phosphate can be seen as the sum of two reactions, as shown. Since the standard free energy change, the delta G0 for the overall reaction, is minus 3.7 kilocalories per mole, and since the hydrolysis of ATP is exergonic, generating minus 7.3 kilocalories per mole, we can calculate the standard free energy change for the phosphorylation of fructose 6-phosphate to be plus 3.6 kilocalories per mole. The enzyme's properties include that it is biologically irreversible and that it is allosterically regulated. The list of allosteric regulators that can inhibit or stimulate phosphofructokinase includes ATP, ADP, adenosine diphosphate, and AMP, adenosine monophosphate, as well as citric acid and fatty acids. Once you know where these molecules are produced or consumed in cells in respiration or other metabolic reactions, think about how and why each of these effectors works on this enzyme. Allosteric regulation of enzymes occurs at major biochemical pathway branch points, often ones catalyzed by biologically irreversible enzymes. Here's an abbreviated view of stage 1 of glycolysis. The first branch point is at the hexokinase step, shown here with a blue dot. The decision here is to either let glucose be metabolized for energy or leave it free to circulate to other cells. The next branch point is the reaction we were just looking at, catalyzed by phosphofructokinase. Here, allosteric regulation will mediate the decision to either use glucose for energy, that is, for glycolysis to proceed, or to store the glucose as glycogen, or possibly even convert it to other sugars like ribose or deoxyribose, which happens, of course, depends on cellular needs. Remember that allosteric regulation can cause a change in the steady state of a pathway. In the next reaction, the enzyme fructose diphosphate aldolase splits the 6-carbon fructose 1,6-diphosphate into two 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. As you can see, the reaction is very endergonic to the tune of plus 5.7 kilocalories per mole. Another enzyme, triose phosphate isomerase, catalyzes the isomerization of DHAP to glyceraldehyde 3-phosphate, or G3P, another endergonic reaction, with a standard free energy change of plus 1.83 kilocalories per mole. The net effect of the two reactions seen here is to split fructose 1,6-diphosphate, a 6-carbon sugar, into two molecules of glyceraldehyde 3-phosphate, two 3-carbon carbohydrates. Added together, these two reactions occur with a total standard free energy change of plus 7.53 kilocalories per mole. These two reactions then would seem to be a very endergonic barrier to glycolysis. But are they? To answer this question, we need to know the standard free energy changes for the two reactions, as I've shown you, and we need to measure the cellular concentrations of reactants and products for each reaction. Here are some actual concentrations as measured in rat liver cells. We can intuit something about cellular energy needs and the two glycolytic reactions just from these concentrations. What do they tell us? Clearly, the overall product, glyceraldehyde 3-phosphate, is at much lower concentration than either fructose 1,6-diphosphate or dihydroxyacetone phosphate. Can you infer something from this? We'll discuss this a bit more in class. Let's summarize the first stage of glycolysis. A 6-carbon carbohydrate has been split into two 3-carbon carbohydrates. A total of 2 ATPs have been consumed here. There have been no redox reactions so far. Remember that the first of them occurs in the second stage of glycolysis. And we've seen two allosterically regulated enzymes, both of which in this case happen also to be biologically irreversible. And that brings us to the end of this presentation. Next up, the second stage of glycolysis.